Hello everyone, welcome to a short video to finish the innate immunity or the non-specific immunity. We've talked about some chemicals you can throw at some attackers, and we've talked about just your first line of defense, that it's just your integument or your outer covering. Now we'll get into a little bit more specific because we're going to talk about inflammation, which is a pretty sophisticated response designed to protect you from invading pathogens, and fever, both of which are still non-specific. They're still non-specific, but they're really going to help you out. So to test ourselves, we'll take these 7 chemicals on the left and we'll decide which of these 4 things, A through D, that they do, with one of them actually going to do 2 of these things. So prostaglandins, keep your eye out for. They're so important. Prostaglandins are actually going to do 2 of these things. So you'll hear about prostaglandins pretty constantly as we walk through, talk about the innate immune system. So innate, adaptive, split right down the middle. We're about to finish innate in this module, and then we're going to move on to adaptive. Adaptive or learned is cell-mediated and antibody-mediated. It's very, very specific to one pathogen at a time, one invading microbe, one bacterial species, one virus. COVID-2, anybody? COVID-19? But the innate, which we're finishing up, is just completely non-specific. So we talked about your first line of defense, your integument, your mucosal membranes. We talked about sloughing off material and getting pathogens out of you. Talked about throwing compounds just randomly at pathogens. Antimicrobial proteins, iron-binding proteins, complement, very non-specific. We've talked about your macrophages and your neutrophils going out, and your natural killer cells also, and phagocytosing or eating up all these pathogens, but non-specifically, just eating first and asking questions later. Now we're going to finish off with 2 things. One, the inflammation response, down here number 3 at the bottom in green, and then fever. So let's start with the inflammation response, which is very, very important. What triggers it? Any sort of infection, which could be, let's say that your integument fails you and it breaks, so you cut your integument, which is shown at the top. Tissue injury, but it's a slice right in the integument with a bagel knife. Burns, other ways for infection, scrapes for infections to enter your body. So you're going to talk about the 3 parts to the inflammation response. We're going to 3 parts, and they are going to be dispose of the invading microbes first, because they'll kill you if you don't. Keep them from spreading, which will really kill you even quicker if they're allowed to multiply. So 1 is going to be about cleaning up the tissue where they're at. 2 is going to be keeping them from getting in the bloodstream and going septic. If you go septic, that means you no longer have an infection at some site of a cut. When you go septic, it means those bacteria or pathogens at that site of a cut are flooding your entire body through your bloodstream and your lymphatic system. And you don't want that. And number 3, you're going to facilitate via inflammation. You're going to facilitate tissue repair. You're not going to do the tissue repair. That's chapter 5. That's the integument. The integument's got all these jobs to do. But you're going to facilitate it via the lymphatic system and via immune function of innate immunity. First thing you're going to do, 3 parts to the inflammation response. We just did 3 roles. Now there's going to be 3 different parts to it. And the first is going to be to vasodilate and increase the permeability of capillaries. That's going to allow for quicker healing. It may seem weird at first, counterintuitive. Why am I blowing up and getting red and painful and swollen and hot in this tissue? Well, because you are vasodilating. You're opening up the blood vessels and you're increasing capillary permeability. That's allowing all the healing factors to enter the tissue. It's allowing your weapons to enter the tissue to do battle, as you can see from this image, with the folks who just came in, the microbes that just came in. So that allows you to do step number 1, the vasodilation. And the increased permeability allows you to put more chemicals in the area. And we'll talk about inflammatory cytokines. We already have histamines, prostaglandins, leukotrienes, all those great things. And then it's also going to allow you to emigrate via diapedesis. Do you remember diapedesis? Emigrate the phagocytes from the blood into the tissue to allow phagocytosis to occur. It'll be really important. And then tissue repair, you're facilitating it by bringing all the materials to the integument. And then the dermis and the epidermis will get to work. They know what to do. They know how to fix themselves. But the lymphatic system and immune function is all about bringing those materials to the dermis, to the epidermis, to facilitate and speed up tissue repair. Deep wound healing. So let's talk about that vasodilation, that increased permeability of capillaries. It's really important. If you remember back to hemostasis, not homeostasis, hemostasis or blood clotting. Remember, the first thing that happens on vascular injury is you spasm and you contract your blood vessels so that you don't lose more blood. But this is tissue injury that we're talking about. So let's just say not quite vascular, but a little bit superficial to that. You get tissue injury, cut, scrape, burn. Pathogens are flooding in. Your response is to actually dilate. And I'll take you to this spot where they show the precapillary sphincter contracted, and that's closed. This sphincter is closed. What do? You simply open it. You expand the muscle. You relax the smooth muscle right there. And you open the sphincter. And once it's open, blood is free to flow through all this capillary bed. And if this is in the tissue, this is going to bathe the tissue. This dilation, this vasodilation is going to bathe the tissue with all kinds of great stuff inside of blood and bring more blood to the area, which of course is going to cause redness because more blood is red. So if you bring more blood to an area, it's going to get redder. Heat, because your integument usually isn't super hot. It's usually several degrees cooler than your core, but now you're flooding it with 38-degree blood, so it's going to warm up. And then edema, because you're putting more blood in the area, and do you remember when you put more blood in the area, you get more tissue, more fluid pushed out via bulk flow out of the typical continuous capillaries. So you put more fluid in than you pull back, that will allow that tissue to swell, which is edema, which is where the swelling comes from. Okay? And you're also going to make it easier at the bottom here, easier if you open up those capillaries and open up those intercellular clefts and open them up. It's going to allow for diapedesis to happen more easily, to get your neutrophils, to get your monocytes, which will then become macrophages, into the tissue. Remember, monocytes become macrophages. So here's a look at this increased permeability. Here is a very simple monocyte. I'm drawing a monocyte inside the blood, and then out. So B for blood would be above. These are endothelial cells lining the capillary. The intercellular cleft, I'll draw an arrow at that. And here are all the invading pathogens that are going to make your life miserable. So what are we going to do here? Well, we are going to open up the space between the endothelial cells, right? Therefore allowing us to move that monocyte through that larger intercellular cleft. So through the larger intercellular cleft into the tissue in order to do battle. And what chemicals, what cytokines, what inflammatory cytokines allow you to do this that are released upon tissue injury? Well, histamines, bradykinin, prostaglandins, leukotrienes, and the complement system proteins are the 5. And you should have a rough understanding of that. What I also want you to pay attention to is a couple of times that basophils come into play. And I am going to make a point of this because I think it's important. Basophils. Why is that important? Let's go back, way back to Bio 40B. And we looked at blood and this breakdown of blood. And we went through, okay. And we went down to the basophils as part of the white blood cells. And basophils are the least popular of them all. There's only less than 1%, really. But we came over here and we said, okay, neutrophils, we gave them Pac-Man, phagocytosis. They eat things. Eosinophils, they mostly eat things. Monocytes, big time, become macrophages and just go wild. Lymphocytes are the brains of the operation, and that's going to be adaptive immunity coming up next module. But look at this. For basophils, what did we put? We put the fire truck. We said when a basophil gets to the site of infection or the site of tissue damage, it acts like a fire engine. It just hoses down the tissue with inflammatory cytokines, with histamines, with prostaglandins, with complement protein, with leukotrienes. Those are the things that come out, and bradykinins that come out of basophils and hose down and cause the effect of vasodilation, relaxing those sphincters, vasodilation, and increasing that space between the endothelial cells, increasing the intercellular clefts so that more chemicals can come out and more white blood cells can come out to enter the tissue. And you're going to see, I already pretty much covered this, but the redness, the pain, and the swelling are going to come from just more blood put into the air. Where does the pain come from? Well, you have nociceptors. Nociceptors are the pain receptors. And they would get dinged if you had something chemical, some noxious chemical, come through and bind. So this microbe may give off a noxious, painful chemical, and it may bind to this nociceptor, which will run right to the central nervous system and tell you, ow, this hurts. This thing that's entering is releasing chemicals that hurt. Or you could have maybe more of a pressure sensor nociceptor, and the edema of the tissue, this tissue here starts swelling out and increasing in size because of all the extra water. And now, lo and behold, by this little kludgy drawing that I'm trying to show you, the tissue now is impinging on this nociceptor, and it's pushing on it, and you're sensing pain that way. So edema and pressure, it should be fairly straightforward. An edemic tissue is highly pressurized with all that fluid, and it starts to hurt. There's nowhere for that fluid to go, so it starts pushing on some of your pain receptors. Or the bradykinins also too. So not only could it be pressure, not only could it be the microbe toxin, but inside this tissue, your program, one of those cytokinins that you're going to be releasing from the injured tissue is bradykinin. And bradykinins are pain molecules. They bind to pain receptors. They let you know that this tissue hurts. It's evolutionarily important to pay attention to a tissue that's infected. That's what the pain is telling you. Pay attention. Don't pretend it's not there. You might get infected. You might go septic. You might die. Pay attention to it. Maybe take a look at it. Maybe clean it up. Maybe put some Neosporin on it. Maybe go see a doctor. Do something, and that's what the pain is trying to tell you to do, is not ignore it. So we have talked about the inflammation and the emigration, and here is, so number one we've covered pretty heavily. Number two, the emigration is right in front of you in this image. Here comes your monocyte out to become a macrophage and gobble, gobble, gobble. Macrophage can just ingest hundreds and hundreds of microbes. And here's your neutrophil squeezing out. And this is the diapedesis. This is the diapedesis. And I'll go back just to review. It will be in the test yourself, but don't stress it too much. This is from 40B. But the selectins, when the tissue is injured, these molecules called selectins, I'll draw it as a squiggly, these selectins come and they implant themselves into the wall of the capillaries. And what do these selectins do? Well, they're sticky. And on the side of the neutrophil, I'll draw one more neutrophil here. On the side of this neutrophil here are integrins. And those are proteins that are also sticky. And so the integrins sticking off of the neutrophil grab onto and stick like Velcro. Stick onto the selectins on the capillary wall. It slows the neutrophils and the monocytes down. It marginalizes them by putting them to the edge of the capillary, and it just makes diapedesis that much easier. And the slowing down is important. Picture a perfectly smooth concrete floor, super smooth, polished concrete floor, and you have a baseball, and you roll it on that concrete floor. It's just going to roll forever, and roll and roll and roll. Now take that concrete floor and cover it in Velcro. And then take your baseball and wrap it in Velcro. The one side of the Velcro on the floor, you could think of like the selectins, if you wanted to. The other side of the Velcro wrapped around the baseball, you could think of the integrins. Now roll that ball, and it's going to catch and grab onto the Velcro and slow down. And you can think of it that way if you want. I have no idea if you want to think of it that way, but you could if you think that would help you to understand that facilitation of diapedesis. And then standard phagocytosis, and you should know this. We've just seen it so many times. Here is the microbe. The macrophage, or whatever this large phagocyte is, could be a neutrophil, could be a macrophage. It is moving by positive chemotaxis. Why is it moving this way towards the microbe? Well, because the microbe is releasing chemicals. It's releasing toxic chemicals around it, signature chemicals. And you are moving in the direction where the concentration gets higher and higher, because the concentration of these chemicals is going to be higher around the microbe, and it's going to drop off as you move further from the microbe. So the phagocyte is going to start at a low concentration and keep moving so that it's increased, so that the concentration that it sees of microbial toxin is increasing all the time. So it gets closer and closer to the microbe. And then the pseudopods are these two little fingers that stick out. They surround, this is a form of endocytosis. They surround the microbe and they phagocytize it, or they eat it via endocytosis. And you could get into receptor mediated. We could talk about a clathrin coated pit. We won't right now. That was more 40A. Let's just understand that it's put into a vesicle called a phagosome, and then that fuses with a lysosome. And we know what's inside lysosomes. Nasty, nasty, nasty chemicals. The lysosome fuses with the phagosome. The two membranes fuse. The contents mix. That makes a phagolysosome. Just combine the two. And there you have digestion with all these nasty chemicals shown in red, digestion of this microbe, and you take apart this microbe and you kill it. And you're left with a residual body, but don't kid yourself. Next module, we're going to talk about something really critical that happens right here. MHC molecules, major histocompatibility, try that again. MHC, major histocompatibility complex molecules. So unbelievably important. They're going to come along and they're going to process and present these antigens. The MHCs are going to dock into the cell membrane, and they're going to take this little antigen here and they're going to present this antigen to the outside world. And that is the bread and butter, the foundation, the heart, the core, the essence of adaptive immunity is the ability to present that antigen outside the cell. And we'll see that in the next 3 modules. And then to finish up, you would simply just facilitate tissue repair by bringing the chemicals. You know what's going to happen. Which cells in the dermis, in the connective tissue, remember the dermis is dense irregular connective tissue, the fibroblasts are going to get to work. And what do fibroblasts do? They build 2 of the 3 components of connective tissue. They're going to put the fibers out and build the fibers, the collagen fibers. They're going to span this gap, by the way, and contract and sew it together. And they're going to lay down chondroitin sulfate. I should do it in beige, but they're going to put down chondroitin sulfate, which is a good matrix, or hyaluronic acid, whatever the matrix needs to be of the connective tissue. That's going to fill in the missing spaces. The collagen fibers are going to contract, and then the epidermis will get to work. Remember, this is a stratum basal. It will go ahead and go through mitosis, and it will rebuild this layer to connect and bridge with new mitotic cells. And then these cells are stem cells, and they're going to build upward. Right? They're going to build upward into the stratum, in the other strata that are above it. Okay, so they're going to build up in the stratum spinosum, and that's going to build up into the stratum granulosum. I won't go too far, and up into the stratum lucidum and the stratum corneum, and upwards you go. These cells are going to build apically and rebuild the tissue. Wound healing, chapter 5, way back. Right? And that leaves us just with fever. And how does that work? Well, the macrophages that are attacking right here, these are your macrophages. Macrophages attacking. As they do their job, they release prostaglandins, right? They release prostaglandins, and they release interleukins. It's really fun to say, interleukin. And the prostaglandins and the interleukins, in heavy concentration, do migrate into the blood, and they are passed down the bloodstream. And you remember circumventricular organs of the central nervous system of the brain, really, including the hypothalamus that is allowed access to the blood. Not many brain organs or parts of the brain or parenchyma of the brain is allowed access to the blood. It gets CSF, which is the filtered version of blood. But the hypothalamus gets to see everything, and it's allowed access to the blood. And it senses high interleukins and high prostaglandins when you're under attack. What do high interleukins and high prostaglandins in the blood do? It triggers the nuclei, the hypothalamic nuclei, to reset the body's thermostat, to bring up the entire temperature of the body up 1 degree, 2 degrees, 3 degrees Celsius, 4 degrees Celsius. When I was young, I unfortunately learned what 108 in Fahrenheit fever would do. My uncle died of leukemia, and he hit 108, and that was the end of the road. So you want to definitely reset. Why? What's the benefit of resetting temperature higher? Well, just a couple of degrees Celsius will kill several different invading pathogens. It'll just kill them. But it will also speed everything up, and metabolic processes increase in speed when you raise the temperature. So you just increase the temperature, things happen faster. Okay? And so you'll heal faster, and you'll kill off some pathogens too. And the more you're infected, the more you need to bring that temperature up, and the dicier it gets. And you really do want to stop if you get too high, because then you're really starting to denature all your proteins. So if you're talking about raising it to 41C, 42C, you're going to start to majorly denature the proteins in your body, and that's when you're going to really feel very, very terrible. Okay? But I'll go back. It will increase. I'll go back just one slide. Increased temperature will increase anabolism. And I do want to take a moment just to refresh your memory that metabolism is 2 parts. Anabolism is the building. Catabolism is the taking apart. So you're speeding up the building or the anabolism. And that's it. let's go ahead and take a look. Bradykinins are going to be good at A, inducing that vasodilation and the permeability of the capillaries. Interleukins are going to induce fever. Those and prostaglandins, so I'll just put that right here for prostaglandins, are going to induce fever at the hypothalamus. But prostaglandins are also really good too at inducing that vasodilation and increasing capillary permeability. Leukotrienes also induce vasodilation and increase capillary permeability. Selectins and integrins, you're not responsible for this module or for this unit or for this quarter because it was 40B, but I just threw it in there because I feel proud about what you learned and bring it back and tie it all together. What do? They facilitate diapedesis. They make it easier. They slow down those neutrophils. Complement proteins, they help to opsonize pathogens for easier phagocytosis. Okay? And they also induce vasodilation. So A is the one you need to know for this module, that complement for this quarter, that in this unit, that complement proteins induce vasodilation and permeability. But remember, they also, from previous modules, they also opsonize pathogens for easier phagocytosis. And histamines, of course, so important. So important that you take antihistamines to try to cut down on the swelling sometimes. That swelling in the respiratory passage is mucus and a runny nose like crazy. And so antihistamines are trying to bring it down because you're secreting histamines like wild to try to swell up tissue and bring all those healing factors to it. Okay, that's it. End of innate immunity. And let's go to a very general module on adaptive overview of adaptive immunity. Then we'll go into cell-mediated and antibody-mediated immunity and really get into the nitty-gritty. Thank you so much for watching. Let's get into adaptive immunity.